Good afternoon and welcome to the third webinar in our series on solid form informatics. It is my pleasure to introduce my colleague Dr. Eleanor Pidcock, a senior member of our Research and Applications Group, to you today. She leads the Crystal Form Consortium, which has been instrumental in developing the tools of the Solid Form Suite that you are going to hear about shortly. Eleanor.
Thank you very much. Yes, welcome to our third webinar. As has been said, I'm going to be talking about assessing polymorphism with the CSD Solid Form Suite. Okay, so I don't think I need to introduce polymorphism to this audience, and so I'm going to skip over this fairly quickly. But polymorphism is the ability of a solid material to exist in more than one form or crystal structure. And what I'm showing here is the two polymorphs of the tonavir. And you can see that the molecular conformations are very different, the intermolecular interactions are very different, and the crystal structures are very different. And it's the different physical properties that you get from these different crystal structures which are of quite high importance in the drug development sector. So, for example, you can affect your drug efficacy by the differences in solubility and dissolution rates between polymorphs. Processing a drug can be affected by the polymorphism, the morphology, for example, or the mechanical properties of the polymorph. And so it's become apparent over the last couple of decades, I think particularly with the events with ritonavir, that it's very important to understand that polymorph landscape. You have to understand the risks of developing that solid form such that the drug product can be manufactured and delivered with consistency. So we at CCDC have been very interested in understanding intermolecular interactions for a very long time now, as shown by these publications here. Really, looking at the parameterization of them, understanding them energetically, etc. But it wasn't really until we got involved in the Pfizer Institute for Pharmaceutical Material Science in 2003 that our understanding of how the roles of intermolecular interactions and polymorphism really impacted on drug development. This Pfizer Institute was a collaborative research venture between the Departments of Chemistry and Material Science at Cambridge University, CCDC and Pfizer Global R&D, and they had a lot of blue sky research projects undertaken in areas of strategic interest to Pfizer, so looking at co-crystal design, for example, or crystal structure prediction. And our role in that institute was twofold really, is to provide software tools that could bring database-derived knowledge easily to the chemists working on their project, but also to try and use our database knowledge to help with that idea of risk assessment for polymorphism. And our involvement in that institute was really very valuable. I mean, to have an industrial partner bringing that focus on real problems as well as giving us a strong lead on what would help address them was really very, very, very valuable. And so we decided to set up the Crystal Form Consortium to continue that dialogue with scientists from the development sector. And this is a consortium of about 11 companies from pharmaceutical and agrochemical businesses. And we set that up in 2008. And really it's to continue with that development work that we started with Pfizer. The goal is to develop software tools that allow those development scientists to make high quality decisions about their solid forms with less risk. And really right from the beginning of the consortium, the questions that they were asking us were very similar to the conversations that we've been having with Pfizer. It's really that idea of can we understand the risk of polymorphism, for example, with our solid forms? Can we address questions such as, have I done enough screening experiments? Did I leave my slurries for long enough? Should I be worried that there is a more stable form still out there? And really that's what we've been working that brings me to the Predictive Analytics concept that Colin introduced in his webinar. And it's this idea of can we estimate future behaviour by looking at data that's been collected in the past? Can we estimate future behaviour with reasonable certainty? Another way of putting that is can we use those millions of interactions that we have in the Cambridge Structural Database to predict outcomes in the solid state? can we comment on the likelihood of polymorphism? Can we enable a judgment about stability, for example? And so this is really what we've been working on with the Predictive Form Consortium over the last few years to develop a software tool that can start addressing some of these questions. And we think we've got something now, and that's what I'm going to share with you today. So this is the output from our software tool. What I'm showing you here is a solid form landscape. It's a particularly complex example, this one. But over the next few slides, as I go through the steps that we use to calculate one of these, I hope to show you that it's not hard to calculate these types of landscapes, and the information that you can get from them, from this landscape, will allow you to assess quickly whether your solid form is likely to be stable, whether you may have issues with stability, sort of investigate what the other polymorphs of the structure might look like. Okay, so I'm going to go through the steps of a calculation. I just want to show you really how straightforward these calculations are. But I also want to show you that I think many of the steps that are involved in calculating one of these solid form landscapes really mimic the type of thought processes you probably go through anyway when you're looking at a new candidate. When you're trying to think about what functional groups might be interacting with each other, what might be a stabilizing interaction, what might be an interaction which is of less importance, and trying to understand what might make a polymorphic structure, how a polymorph might look. Okay, so our first step is to load in a prediction molecule. And so you can load in either a 2D diagram or a 3D crystal structure. And the example that I'm going to talk about is lamotrigine. And I'm going to show quite a few of these screenshots of the wizard interface. So we've got a wizard that guides you through these steps, and you'll see quite a few of these screenshots from that wizard. So the first step, once you've loaded in the molecule, is that the donor and acceptor atoms are identified in the molecule, and they're listed on the right-hand side. And if you click on one of those donor or acceptor atoms, it highlights it in the sketch of the molecule. The next thing we do is define functional groups that encapsulate the chemistry of those donor and acceptor atoms. Now the tool already has a library of about 90 functional groups, and it will try to match up its library of functional groups with the donor and acceptor atoms that are present. You can also sketch your own functional group definitions, and you can add those to the library, or you can edit the existing functional group definitions. And so it can be very flexible in how those functional groups are defined, and you can ultimately control the functional groups which are in the library. So once we have the functional group definitions defined, we now have the basis of our search that we are going to pass to the database. So those functional groups are then submitted as a database search. And so any molecule that contains at least one of those functional groups will be returned as a hit and put into the database. And as the search proceeds, the interface gives you an indication of whether you have enough data or not, so whether you have enough structures in your data set covering those functional groups. And so it's trying to help you all the way through decide whether you have enough data or your data is good enough quality. Often what you will find is if you're searching for a very common functional group, so for example a hydroxy group, is that you'll end up with tens of thousands of structures. And in general we don't want to give that much data to the statistical model, and so it's a good idea to reduce those data sets down. And there's a really nice feature for the data set size reduction. Behind this slider down here at the bottom of the window here is an algorithm which keeps the most similar structures to your target structure. That's where the functional groups in the data set molecules are most similar to the functional groups in the target molecule. It keeps those for longest. So although you end up with data set sizes of, say, 200 or 300 molecules, they are the most relevant molecules in the database to your structure, and so you end up with a highly predictive data set. And it might be small, but it's very predictive. Okay, so the next step is to run the hydrogen bond analysis. And so this is when the algorithm is looking at the data that it's collected and extracting the useful information from it. So what we have here, again, this is behind the scenes. You don't see any of this, but what we have here is the output from that step of the calculation. So we have our ref codes here. We've identified the donors and acceptors, and we have whether the hydrogen bond exists or not. And so this is very similar to sort of frequency of occurrence type search that you do in Conquest. But this tool is much more sophisticated than that. So rather than just looking at the frequency of occurrence information in order to make its predictions about whether a hydrogen bond will be observed in the target structure, it's also pulling out these explanatory variables, so information about the explanatory variables. So is the functional group sterically hindered, for example? Is it close to an aromatic group? Is there a lot of competition present in the molecules? And so by taking this information, we can start to build an understanding in the statistical model of when a hydrogen bond will occur or not. And so this information is extracted, is fed through into the predictive steps. Okay, so that brings me back to this predictive analytics step. So we now have the data, and now we have to analyse it and fit the statistical model. And so I'm not going to go through this slide in a great deal of detail, but really what I'm showing here is we are wrapping up all of our explanatory variables, so the steric density, the aromaticity, competition, in this function Z here. And we're plotting our data. So we've calculated our Z values for all our structures, and we're plotting against whether the hydrogen bond doesn't exist, so Y equals 0, or whether the hydrogen bond does exist, Y equals 1. And what we can see in this example here is that there's a good separation between the explanatory variables for structures where there's no hydrogen bond observed versus structures where there is a hydrogen bond observed. And so this is going to be a very predictive data set. And so using logistic regression, we fit our characteristic F-shaped curve to that data, and that's going to allow us to read off a value, our propensity for hydrogen bond formation to occur in our target molecule where we know what the values of Z are. And that's exactly what we have done here. This is the first piece of output really that you see from the tool in the wizard. And so this is what we have. We've got our donors here, the donors in the Lamotte gene, with the acceptors in the Lamotte gene, and the propensity. So these are the values that we've just read off that curve that I showed you. And you can see that we're predicting that there's some really good interactions possible in the Lamotte gene. So if you have a high value here close to 1, that's indicative of a strong and likely interaction. Values approaching 0 are indicative of unusual interactions and possibly could be a cause for concern. But in the case of the Lamotte gene, we're predicting a couple of really quite strong interactions. And if we look at the solid state, the crystal structure for the Lamotte gene, what we find is indeed this interaction that we've predicted here is occurring. We've got these NH2s donating into the aromatic nitrogen, and also the second interaction here also occurs. So again, NH2 donating to the other aromatic nitrogen. And so if this were a drug development scenario and we had this sort of prediction matching up with our solid form, we'd be feeling fairly confident that what we had was a good candidate. So we gave that version of the tool to a Crystal Form Consortium, and they came back to us fairly quickly with some feedback about it, and that was that the tool, that table I just showed you, was very useful in identifying whether the candidate that you were currently looking at was likely to be stable, or whether it had some unusual hydrogen bonds in it which might indicate that it was possibly not the most stable form. But the piece of information that was missing really was how you could compare that structure with whatever else was possible. So although we have this table here that enumerates all the possibilities of the hydrogen bond combinations, it's very hard to sort of understand how you could put them together to build up another possible structure. And then it's even harder to understand what the stability of that structure might be in relation to the solid form that you have. But if we have an understanding of, say, how often a functional group is likely to participate, then we can build up pairings of functional groups to generate possible structures, and we can rank them in terms of how satisfied those functional groups are. And so once we have that ability to build possible hydrogen bond combinations and understand whether they're satisfied or unsatisfied, we can sort of build up this landscape of possible structures that you can view your target structure in. And so again, this is another example of a predictive analytics. We've gone back to the database and we've determined, again using logistic regression and statistical modelling, propensities for functional groups to participate no times, one time, two times, etc. And so now we have this model, we can calculate those landscapes and rank structures in terms of the quality of the hydrogen bonds and the participation of the functional groups. And so that's really what I'm showing here. I'm going to go through some examples where I can show you what the information this tool gives you. But just first, I just want to show you the chart to make sure that you have a clear idea about what we're plotting. On the x-axis we have the mean propensity, so that's the quality of the hydrogen bonds. Those are the propensity values taken from the table. And against that, on the y-axis, we're plotting participation. And so this is how satisfied the functional groups are. Are they participating in hydrogen bond in line with expectations from the database? And so if we have poor participation and poor propensity, we're up in this area of the chart, and that would signal that you've possibly got some stability issues with the solid form. Whereas if we're down in the bottom right of this chart, then you probably have quite a good stable form. You've got good, strong hydrogen bonding, and the functional groups are participating at the level they're expected to. Okay, so I'm going to go through some examples, as I said, and show you what the information you can get from this tool is. We've kept the examples really quite easy, I think, for this presentation, just so you can concentrate on the information that you get from the tool rather than sort of drowning in the complexity of the donors and the acceptors. So our first example is omeprazole. It has one donor and 5 acceptor groups. And if we run that through the hydrogen bond propensity tool and plot the solid form landscape for it, then what we see is a chart like this. So our grey triangles are all the possible combinations of the donors with acceptor groups in omeprazole, and our target structure, so our crystal structure, has been highlighted in pink. So this spot here has the hydrogen bond combinations which are present in the solid state. And so immediately we can see that we're in the stable part of the chart. We've got really the optimum outcome for propensity and participation. And we can see in the 3D structure that we have this nice hydrogen bond between the donor and the best acceptor. And so if this were a drug development scenario and you had a solid form that gave you a chart that looked like this, you'd be feeling pretty confident that you had something that was quite stable. You could match up with your experimental results, but you'd be feeling quite confident that there weren't any issues with the hydrogen bonding or stability of this compound. If we go to another polymorphic drug, this is piroxicam. There are a couple of polymorphs of this in the database. If we take one of these structures and again run it through the hydrogen bond propensity tool and calculate the solid form chart, solid form landscape, then what we see is something that looks like this. Immediately this is much more worrying than the previous chart, I think. You can see that this solid form is on the left-hand side of the chart in the unstable area. We don't have good propensity and we don't have particularly good participation. And also what you can see very quickly is that there are much better candidates available, but there are better ways of combining those hydrogen bond donors with acceptors. And again, if we look at the solid state for this polymorphic piroxicam, you can see it has an intramolecular interaction, but there are donors and acceptors, good donors and acceptors that aren't participating at all. Hence, it's viewed as being a less stable outcome. And again, if you were in this situation in the development scenario, then this would be a warning sign. You probably need to go back, look at the experimental results, see if there are other polymorphs out there which are likely to be more stable prospects. As I said, it was a polymorphic structure, and there is another polymorphic piroxicam in the database. And again, if we run this through the hydrogen bond propensity tool, we immediately see something that's a little bit more reassuring. Again, our target structure, the crystal structure of this polymorph, is over on the right-hand side of the chart, indicating that we have something, a good set of hydrogen bond combinations and functional groups participating at a level that's expected. And we can see immediately, again, we've got donor interaction with best accepted, and the intramolecular interaction remains. And then if we go back to the thermodynamic data on these polymorphs, we do indeed find that it's the second polymorph here that is the most stable form. Okay, so another example. This is temozolomide. Again, quite a simple molecule, one donor, 5 acceptors. But we're going to run this through the hydrogen bond propensity tool and have a look at what it looks like. So we've done the calculations here. I'm showing you the table. So we've got our donors with acceptors with propensities. And you can see that this interaction here, the carbon-moil dimer, is predicted to occur with quite high likelihood. And indeed, we see it in the solid form. It's this nice dimer interaction. If we look at the solid form landscape for temozolomide, we see a chart quite different from those that we've seen before. So our target structure, the crystal structure, is shown in pink, and it's in quite a stable part of the chart. It's had some of the best propensity values, and the participation is quite good as well. But what we're seeing in this chart here is that the pink spot is surrounded by grey triangles. So this is indicating that although the solid form that we've looked at has some good hydrogen bond combinations, there are also other competitive hydrogen bond combinations out there. So there's other ways of pairing up those hydrogen bonds which look about as good as the solid form that we have in front of us. So I'm just going to focus in here on other data that's given to you through this tool. This is really the participation data. So this is showing you which hydrogen bonds are present in the structure that's indicated by the triangle or spot, and how the functional groups are participating. And so we can see quickly that the NH2 group is participating once, which is an optimum outcome in this case, which is why it's colored in green. The nitrogen acceptors aren't accepting, which is the optimum outcome for them. Our carbon-moil oxygen is accepting, which is good. But the carbonyl oxygen is not accepting, it's not involved in hydrogen bonds. So in this solid state, we can see that this carbonyl group is not involved. If we have a look at the other points surrounding that pink spot, we can see what those potential other structures would look like. So if I click on this spot, it changes what's highlighted in this chart. So now we can see that in this structure, the NH2 group is donating twice. The nitrogen groups aren't accepting, but the carbon-moil oxygen is accepting, and the carbonyl oxygen is now accepting. Again, if we travel around these spots, we can see in this structure, again, the NH2 group is donating twice. One of the nitrogen atoms of the ring is now accepting. It's not the optimum outcome, but it's still worth considering. Our carbon-moil oxygen is accepting, and the carbonyl is not accepting. Again, we keep moving around. You can see again we've got a different combination of hydrogen bonds here. This time in the database, nitrogen is accepting. And so if we look in the database, there are polymorphic structures of temozolomide available, and I can show them here. This is the most stable form. You can see we've got our carbon-moil dimer. We have NH2 donation to the carbonyl group, and NH2 donation to one of the aromatic nitrogens. This one here is the least stable form of temozolomide. So again, carbonyl dimer, NH2 donation to the aromatic nitrogen, NH2 donation to the indazole. And so what we've learned from this is that those hydrogen bonds that were predicted to be competitive with the hydrogen bonds that were observed in our target structure have indeed been found in the polymorphic structures. And so again, if you were in a development scenario and you saw something like this, then it's a warning that possibly polymorphism is going to be an issue, and that it would be useful information to go back to the experimental data that you've collected and to see whether indeed you have found those polymorphs or you have indications of those polymorphs. it's a warning to be careful. There could be issues with this drug compounding, so you need to be careful with that landscape. Okay, so I just want to show you very quickly a sort of neat little function in this tool. Again, this was a suggestion from the Predictive Analytics Consortium, but you can link up that table of the donor and acceptor, the donors and acceptors and propensities with the chart. So if I click on an acceptor in the table, it will light up the triangles in the chart where that acceptor is accepting, or where that donor is accepting, or where you see the interaction. So just very quickly for temozolomide, you can see that I've clicked on where the imidazole is accepting for these points. This is where the carbonyl is accepting. It highlights the different plus points. And this is where the carbonyl oxygen is accepting. And so immediately you can see that carbonyl oxygen interaction, that dimer interaction, is present in all of the best structures that we've predicted. And so that can give you another clue about perhaps what is a stabilizing interaction in your system, and can provide you with additional information when you're perhaps assessing a polymorph or looking for other polymorphs, or in fact maybe if you want to design a co-crystal, for example, other interactions which might be hard to disrupt, other interactions which would be easier to disrupt, for example. Okay, so our last example I'm going to talk about, this is a really very complex molecule. We have 4 donor groups in this molecule, and I think about 6 acceptors. It's really quite complicated. And just looking at the 3D structure of one of the polymorphs, this is Famotidine, these are hydrogen bond interactions, and if you grow out that lattice, you can see the interaction is very complex, and really just understanding what's going on is quite hard in this structure, let alone trying to think about other possible combinations of donors and acceptors and understanding how their stability might relate to what you have. But this is the beauty of the tool, really. We can take that polymorph, run it through the hydrogen bond propensity tool, and see a table like this. So we've got our donors and acceptors and our propensities for those hydrogen bond interactions. And we can see that we've got some good stabilizing interactions present in this polymorph, but we also have some interactions in the mid-table area. Some of them are starting to get a little bit more marginal. But if I just click on those hydrogen bond interactions that are mid-table and highlight the structures that have those interactions in the landscape, you can see immediately that we're not in a particularly good area for those of this landscape. They do look like there's much more stable structures available down towards this way. If we look at another polymorph of Famotidine in the database, then we can see it again immediately. We run it through the hydrogen bond propensity tool. Again, we have 3 nice strong interactions at the top, same interactions that were present in the previous polymorph, but these mid-table interactions are starting to look a little bit better. In general, they have higher propensity values. And again, if I click on those interactions and show them in the chart, what you can see is that we certainly move down to a more stable area. Some of these structures are looking like really very good prospects indeed. And so, without going into too much detail with this analysis, what you can see very quickly is that this second structure looks like a more stable prospect. You can find out this information quite quickly. And indeed, if we go back to the thermodynamic information available for Famotidine, it is this second form here that is the more stable one. Okay, so hopefully I've shown you how the tool works, how easy it is to go through the calculations and how you're guided through those steps. And I've shown you the type of information that you can get from the tool. And I really just want to stress that the tool is really designed to sit alongside your chemical knowledge, the knowledge that you have already about the compounds, and sit alongside the experimental knowledge that you build up. We're just adding to that with our database of millions of interactions and the ability that we have through our algorithms to crunch the numbers. But really, by presenting that quantitative view of the solid form landscape, it really should allow you to understand, to build up that understanding of your candidates through the drug development process. It's also a tool to explore alternatives, and I've not really talked about that in this webinar, but that's the topic of the webinar given by my colleague, Dr. Pete Wood, in a few weeks' time, talking about using this tool for co-crystal design. Okay, so the Crystal Form Consortium continues. We've finished phase 1, but we're about to enter into phase 2. And I just wanted to very briefly show you some of the development work that's going on at the moment, just to give you an idea of the directions that we're heading in. We're working at the moment on a tool which should automate the analysis of multi-component forms using a hydrogen bond propensity tool. So we're looking at how we could automate, say, in silico co-crystal screening, for example, or interaction with solvents. We're also working on a tool that, again, extracts knowledge from the database to apply to systems that don't have very many or don't have your traditional hydrogen bond donors and acceptor groups. So what information can we pull out of the database that allows you to perform a risk assessment when you don't have those traditional bonding groups. Another tool that, or another area that we're working on at the moment is morphology prediction. So we have a collaboration with the Institute of Particle Science and Engineering up at Leeds University, where we're including attachment energies into our morphology calculations. And we're really hoping this opens the door on us being able to interact with surfaces of particles or models of surfaces of particles in a really nice way, perhaps using some of our tools so we can look at and perhaps predict impurity interactions or inhibitory interactions of those surfaces. So hopefully you've seen things that are of interest to you in this presentation. There's plenty of ways for you to find out more information from us or get in touch with us. As I mentioned, we have a webinar coming up in a couple of weeks' time talking about co-crystal design. We have some white papers that are about the solid form informatics tools that we've been talking about. You can request a trial of the Solid Form Suite, so you can try and run this hydrogen bond propensity tool, calculate some solid form landscapes for your target molecules, and you can participate in the Crystal Form Consortium. If you'd like to get involved in helping us develop software tools that will be useful to address the problems that you have, then do get in touch. So that's me finished. Thank you very much for listening. I'm just going to leave you with a reminder of our final webinar in our series. Thank you.